Can everybody see that?
Yep.
Have we got the main presentation up? Everybody got the green screen?
Yeah, I can see it right there.
Cool. Right. Well, thank you very much and good afternoon. Thank you very much, Rita, for the nice presentation. Yes, it's a roll of the tongue, histocompatibility and immunogenetics. I'm so sorry. But this is the field that we work in. And this is what my presentation is going to be around. I'd like to give you a presentation of what's happening in terms of the science behind what we do. And of course, it's going to be around stem cell transplant and the HLA typing. I'm trying to keep it as less heavy than as I can. But of course, if there's anything that you want me to repeat, just pop it in the chat, and then we can talk about it later. So my name is Dario. I'm a clinical support supervisor in the labs, and yes, the first thing I think we need to talk about, and next slide, will be the stem cell transplant. So who needs a hematopoietic stem cell transplant? Well, any patient that has a disease, either a cancer disease or a metabolic disease or a genetic disease that impairs the normal functionality of the bone marrow, of the stem cells, which is basically their purpose is to make red blood cells, white blood cells, and platelets. If the patients have any disease that impairs this functionality, then of course they need therapeutic approaches that can be classical approaches like chemotherapy or radiotherapy, but then of course they not always work. So of course that's when a stem cell transplant is needed. Where do we find the stem cells? Well, you can see there in the screen that the main three sources of these clever, amazing cells are the bone marrow, the blood, and the umbilical cord. So when a donor donates their stem cells to the patient, this is the main three sources where we can take the bone marrow. Now, where you can take the stem cells. Mainly, we say bone marrow because historically, this is where the cells used to be harvested from. But nowadays, we can use also the blood. So the donor is happy, awake, and watching TV, drinking tea. And of course, thanks to this way of donating, of course, the number of donors that will to donate the stem cells has increased incredibly, which is great because not everyone likes needles and cord blood, of course, because we have our bank in Nottingham where we store all the umbilical cord to harvest these amazing cells. So in the next slide, we will see that the outcome of the stem cell transplant is not always as we wanted. In general, and this is a figure that it's all the same roughly in the world. So this is kind of like a figure that describes the situation of stem cell transplant all over the world, not just in Ireland. Roughly, it's a 50-50 chance of survival after 2 years, because of course there's many factors that can impact the transplant itself. So next slide, we will see that around 50% of patients unfortunately can't make it after the transplant. Of course, this is an average. It could be much above 50 or below 50, depending on a lot of factors and how the degree of compatibility between patient and donors. But mainly, the reason why patient unfortunately can make is the relapse. That means that, and this applies to cancer, to blood cancer, the original disease, the primary disease, unfortunately hasn't been eradicated totally before the transplant. So that means that certain cancerous cells can survive and unfortunately can expand after the transplant, creating a relapse. That means that the disease comes back. And this, of course, can be a cause of death. But also we have what we call non-relapse mortality disease cause of death, which could be infections, could be toxicity. A transplant is very invasive, and GVHD. Just to describe very quickly GVHD, I always use my example. In this case, my donor, let's pretend it's Alice. Let's pretend that my kidney is not working, okay? And I need a replacement. So Alice is my donor, the kidney goes inside me. What we need to be scared of here is the rejection, as in, am I going to reject Alice's kidney? With stem cells, it's slightly different because my immune system will be completely depleted because the stem cells themselves are responsible for the immune system. So before the transplant, my immune system will be completely depleted. And when the stem cells of my donor, Alice, thank you, go inside me, what we need to be scared of here is not really rejection, because I don't really have any tools to reject, but it's the opposite. If graft versus host, so it's the graft, so it's Alice cells that go inside me and actually recognize me as an enemy, as in something like, oh my God, where am I? And attacking me. This is graft versus host disease and can be quite severe, sometimes even more severe than the original primary disease. So this is what we need, why we need to make sure that patient and donors are exactly the same, as in we need to trick the immune system pretending that the patient and donors are the same person, at least immunologically speaking. In the next slide, we'll see that the aim of Answer in Ireland is a very amazing charity, which, by the way, you are part of and you contribute to, and we're really grateful for all the work that the volunteers do. Really, we really couldn't do our work without you. Is that we provide the best stem cell donors for any patient. Of course, also we want to improve the transplant outcome, not just finding the donor, but make sure that the patient's outcome, the transplant outcome is the best as we can. How do we do that? Well, first of all, we need to provide the best level of matching, so the best donor that is out there. We also provide post-transplant care, physical, but also psychological and practical needs. So we have dedicated nurses for this. And we lead groundbreaking research. That means that we always lead this field of, we call it HNI because it's easier, with new techniques, and we study new genes, and we lead the scientific committee. So that's what Answer in Ireland Science Department do. So in the next slide, actually, let's go a little bit more in details of what we match between patient and donors. What we match is a group of genes called HLA genes, short for human leukocyte antigen. These genes produce some molecules, and these molecules are great because they are responsible for discriminating what's self, us, from what's not self, the foreigner, the invader, the pathogen. Now, normally these molecules are great because they actually help us discriminating from what's us and what's not us. There are 6 major genes that produce 6 major molecules, HLA A, B, and C, and the RDQDP. Each person has got 2 copies of each, because of course we have 2 parents. So I will have 2 A's, 2 B's, 2 C's, 2 DR's, 2 DQ's, and 2 DP's. So you can see already that how it's starting being difficult finding a match for a patient, because of course we need to match for all these molecules. Now, next slide, please. What do actually these HLA molecules do? Well, I think that these molecules, they look like kind of like the Sentinel-2. So if you see in this comic, we have a cell, a purple cell, and when these cells get attacked by someone, an invader, a foreigner invader, what happens is that they will use the little arms, the HLA molecules, to expose pieces of this invader and call the immune system, saying something like, hey, I've been infected by this one. Can you please go and kill me, all the other systems that have been infected by this one? So the immune system, which is the guy with the helmet, go and alert everyone else, and kind of attacks the foreigner, the invader, sorry. This is what the HLA molecules do. It's a tool to tell the immune system, I've been affected, I've been infected by something, please kill this pathogen. The problem is, this is normally what happens physiologically. In a transplant environment, you can imagine how you have to make sure that HLA molecules are the same between patient and donor. Otherwise, there will be a lot of non-self recognition if there's a mismatch. And of course, you know, the outcome of the transplant might be not great, because as I said, what happens is that the cells transplanted will recognize everything like non-self and attacking it. Next slide, please. So I think in my imagination, HLA molecules look like Lego. So if you look at the next slide, you will see that we have every person having different pieces. Next click. And each of these pieces are different. And next click. The further complication is given by the fact that we all have two sets of these pieces, the bottom 6 inherited by mum and the top 6 inherited by dad. So really, we have 12 pieces of Lego that we need to compare between patient and donor. So we need to find someone out there that has exactly the same pieces of Lego of our patient. And that sometimes, of course, proves difficult. Next slide. Sorry, another one. There will be a lot of clicking here, so you can just click as I speak if you don't mind. So let's pretend that we have Homer here. He has 2 sets of HLA molecules, okay? The green set from his dad and the purple set from his mum. Then again, Marge comes along. She has 2 sets of HLA molecules, the orange from her dad and the red from her mum. Now, of course, Homer and Marge have kids, and what happens is that we will have each kid inheriting randomly each half from each parent. So Bart, for example, will have inherited the green part from Homer and the red part from Marge. And then Lisa comes along and she has inherited the green part, the green half from Homer, but the orange one from Marge. So Bart and Lisa are not compatible. And so on, basically, with the rest of the Simpson family. I do know that not all these characters belong to the Simpson family. Sorry, I just needed a big family to make the example. So if you click again, let's pretend that Bart in this case is our patient. Bart has a disease, unfortunately, that impairs the normal functionality of his bone marrow, his stem cells. So what happens is, first, we will have to find a donor within his family. This is what we do in labs. In this case, Bart is lucky because he has Ralph or Martin. I never remember. I think Martin. That it is Martin. Thank you, Alice. I can see you mouthing. And in this case, these 2 siblings have inherited randomly the same halves from mum and dad. That means that this is a HLA identical siblings, which is without any doubt the best donor that you can have out there. And the transplant in this case can go ahead. If you look, for example, Bart and Lisa, they only share half a haplotype, or we say, sorry, I said something in science jargon, half of their HLA type. In this case, we call them haploidentical, and the transplant can go ahead, but it's quite risky. Then if you look at Maggie and Bart, they are completely mismatched. In this case, the transplant cannot go ahead. Now let's go back into, let's pretend that Bart is not that lucky. So if you click again, Martin is not there. What happened? Oh, sorry, again. There you go. What happened if Martin is not there? Of course, we're not going to let Bart die. We need to find someone out there in the world that has inherited randomly the same HLA molecules from mum and dad. That's why the idea of a register, that's why the idea of anti-Nolan was born. If you click again, we will see that we need to find someone out there who is a matched unrelated donor. So someone in the register, an amazing person who is willing to voluntarily donate his or their stem cells to a complete stranger. And this is exactly what we have. This is the seed of Shirley Nolan that in the late 70s has got the idea of not, you know, she was fighting to not let her son die. And thanks to her, now we have more than 800,000 donors in our register. Now, of course, finding a match unrelated donor is not always easy. You need to find someone that has exactly the same pieces of Lego. So, you know, it depends on the ethnicity and depends on many factors. So it's very important that we do this search in the best and in the most accurate way. So in the next slide, just a step back. How do we do matching people, patient and donors? Well, as you know, we all have DNA inside us, and DNA is kind of the recipe to make us. And we have a lot of cells, and each cell has got DNA. Now, DNA is organized in chromosomes, and basically, if you click at the next slide, to simplify that, I think that this is how, okay, so this is how chromosomes look like. Let's pretend that this is my karyotype. So these are my chromosomes. It's an X and a Y. Yes, I'm a male. Yes. So in my imagination, this chromosome looked like a bookshelf. If you click again, that's it. It's basically a lot of information stored there. And what we actually need to look at in particular is chromosome number 6. Click again, please. In chromosome number 6, click again, is basically book number 6. There is a bit where is written the recipe to make the HLA molecules. So that's what we do in lab. We open book number 6 to look at the recipe to make these molecules. And in fact, yes, if you keep on clicking, there will be, yes, a little bit of the chromosome 6 that we actually investigate to look at these HLA genes. And click again, it's like a little chapter of this book. So if you click, we have 4 main techniques that we use in labs to go and investigate, to read this page of book number 6. I'm not going to go too much in details. There's a lot of techniques that I use out there. I just wanted to say that, for example, the first 2, if you keep on clicking, the first 2 now have been put, they are now obsolete because this field of science evolves a lot. So if we need to keep on up to date with the scientific evolution of the field. So now we use next generation sequencing as our main approach for HLA typing, and it's a really great techniques. If you click again, I will quickly describe it to you. You can, thanks to this next generation sequencing, you can have a high sample throughput. That means that you can do a lot of samples. Scalability, it's very scalable. That means that you can do a small cohort of samples or a big cohort of samples. It's flexible because you can decide how many samples to run. And of course, it's a competitive market, so we chose the best. And if you click again, we can, thanks to NGS, we can deliver HLA typing results in up to 4 days, including data verification and authorization. It's really quick. Imagine that the transplant center send us a blood sample, and in 4 days, we can tell them exactly which HLA molecule the patient or the donor have. So the transplant center we will comment and infer the grade of compatibility between patient and donors. And then, of course, the transplant center will have all the information available to make the clinical decision. We can do a lot of genes in one go. We can achieve a very high resolution. The error rate is very low, and there's various steps, which are probably I'm not going to describe too much into the details, but it's a very great technique, and we're very lucky to have been able to implement it. Next slide, please. The very cool thing about this technique is that you can mix and match samples in the same tube. So before, when I started this job, if you mix and match people's DNA in the same tube, that was called contamination. You have to throw it away in the bin and start again. But with this great new approach of DNA sequencing, you can mix and match people's DNA because they kind of get labeled, barcoded. So for example, let's pretend that sample 1 is labeled in red, and this is me, and sample 2 is labeled in blue, and this is Alice, and sample 3 is labeled in green, and that is Rita's. So it doesn't matter if we mix them together, the software is going to be able to tell you which one is which helps you a lot, because cuts a lot of time, and time is precious. Time is super precious for patients. Transplant center need to have sometimes they need to have the information as soon as possible. Next slide, please. So another cool thing about NGS is that instead of reading just bits of the book number 6, like here, which is a very old way of doing sequencing, just a red bit, with NGS, in the next slide, we will see that actually we read it all. Next slide, please. There you go. We kind of cover the whole page. That means that we are giving to the transplant center much more information, and that means the best donor is selected. Next slide, please. So before, we used to tell the transplant center something like that. Well, yes, this donor is A0201, or maybe 0201L, or maybe 0201Q, or maybe 209, and so on. So it's the majority of labs give an ambiguity that needs to be resolved, but thanks to NGS, in the next slide, we can see that we can give the transplant centers the ultra high resolution, which is great because thanks to this information, they're going to be able to know exactly the match between patient and donor. Another slide, please. One thing I wanted to add is that not only HLA is important when you select your donor, there are other important factors that you need to be selected. CMV, the ABO, the age of the donor, the gender, the state of the disease. But one of the most important in this case is CMV. So in the next slide, there you go, CMV is highlighted. What is it? Next slide. Next slide, please. So CMV is called cytomegalovirus. It's a virus, it's a common virus, part of the herpes family. And you can see that basically in the Western Europe, in Germany, UK, roughly 50 to 60% actually of people have had this virus. Even 83%, worldwide, there's even 83% of people that have encountered the virus. Normally, it gives mild symptoms. You might have even realized that you have had it. But the characteristic of this virus is that when you get it and you get better from it, it kind of goes to latency. It goes to sleep in your body and it stays with you. And it's absolutely fine for a person who has, unhealthy person. But if you are a stem cell patient, that's very important because, of course, if you have reactivation of this virus, it can be life-threatening. So it's extremely important that a cytomegalovirus state gets checked as well as HLA in a stem cell transplant, because when it's matched, that means that the donor is negative and the patient is also negative, it's fine. If the patient and the donor are both positive, that's fine too. But the problem is when there's a mismatch. So if the donor is negative and the patient is positive, it's important. The risk is that there are no cells in the donor that are able to keep that virus at bay, so it could reactivate. On the other side, if the donor is positive and he gets transplanted into a negative patient, of course, that can infect the patient and it's risky. Imagine having a CMV infection at the same time while you're trying to fight your primary disease, the cancer. So it's very important that we also type a CMV, the CMV status. And I think that was the last slide of my presentation. Yes, any question will be, I'd be happy to answer or clarify any. Thank you very much.
Thanks, Dario. I don't know about anybody else, but sometimes I feel like I'm back in school and I start panicking when I feel like I'm in a science lesson, I'm not going to understand it. But with the help of Simpsons, which is definitely one of my specialities, that made a lot of sense to me. And I'm actually thinking with the Lego and the Simpsons, there's things that I think I'll start saying in my presentations that I deliver, that I might deliver things slightly different. I feel a little bit more equipped to answer some questions that we might get from young people around matching and the register, and from the public as well. So thanks so much, Dario.
My pleasure.
There's a couple of questions that have popped up in the chat.
Yep.
So the first one, they're both from Kevin. So if anybody else has any questions, please do jump in. But the first one was, is the transplant process different depending upon how stem cells were donated or harvested?
Oh, that's a good question, Kevin. The transplant process itself is exactly the same, because if either the cells, if they are harvested from the bone marrow, it's just, the way the harvest is, of course, is different. So there will be the bone will have to be pierced, and the cells will have to be harvested this way. And if the donation is through the blood, instead, you basically are attached to a machine called a apheresis machine, and they would just take them from the blood. Ultimately, what you end up with is a bag. Is a bag of cells. So it doesn't really matter the way you harvest the cells, because ultimately it's a bag that gets then reinfused in the patient. What changes though is the way of donating. Of course, one is in general anesthetics, and one, the donor is awake. But of course, the number of cells harvested can change. Usually in the bone marrow, there's a lot, there's many cells. So that's why we'll have to stimulate the donor with an injection 3 days before the donation via the blood to make sure that the cells move from the stem cell, from the bone marrow to the blood, to make sure that we harvest as many as possible. Sometimes, depending on the center, on the transplant center, or the disease of the patient, it could happen that they ask one way of donating rather than the other. But normally with the techniques available now, I would say that either now are fine.
Did that answer it, Kevin? Feel free to come off mute, anybody.
Oh yeah, absolutely.
And ask questions. I'll ask, I don't mind, unless you want to unmute Kevin and ask. But Kevin's next question was, do you think one day, I think this is a really interesting question, do you think one day that it will be possible to generate stem cells in the lab to match a patient rather than taking from a donor? And I'm not sure
I love
this, Dario.
Right. It's funny because A, I love this question, and B, I get it all the time, and it's really, really clever. I would say I don't exclude the possibility that this one day will happen, but we're still quite far from this kind of technology, of course. But when we get there, we will have to find a different job. No, I'm joking. It's, of course, the patient donors compatibility won't be a need anymore. Of course, we will all have to specialize into engineering and bioengineering. But yes, I could see that at some point, maybe we can get there, but the science is not quite there yet. So I still can have a job as a. No, I'm joking.
We might have retired by then, maybe.
No, I think I will be, yes. But if you think about it, recombination and then bioengineering is actually a branch of science that is actually progressing quite a lot. So yes, there is a possibility that you can create kind of bioengineer cells that can be, but definitely we're not there yet.
Thanks, Dario. Another question from Marion. Does the progress in matching reduce mortality from GVHD?
Absolutely, yes, that's exactly what the progress in matching does. So the more we know about the science that lies behind the compatibility and the genes involved and everything, the more we know that there are other genes out there that we can match between patient and donor, the more we're able to reduce the GVHD. Because of course, GVHD ultimately is when the donor recognize the enemy as the patient as an enemy. So if we uncover and discover all the possible genes that can be matched between patient and donors, we will trick the immune system, pretending that actually those cells belong to the donor himself or themselves, the patient themselves. So there's no graft versus host disease. So that actually is the goal. The graft versus host disease curve, we want it to be as flat as possible, as in we want all the patients surviving after the transplant without GVHD. Of course, that's where we're aiming. There's a lot of publication that Anthony Nolan Research Institute has produced towards this, and studies that have identified how better matching and further and more into details matching can reduce graft versus host disease. So yes.
Thanks, Dario. I think I know when I talk about Anthony Nolan in the register, one thing that I'm always really mindful to do is to talk that we're more than just the register. We're all this, or that Dario talked about with the Research Institute. And that we are absolutely committed to improving patient survival post-transplant. And I think that's just fantastic. So it's great to hear directly from Dario as well. Question from Jean here. Why is bone marrow preferred for some patients? Is the choice always based on patients' needs?
Yeah, I think I kind of briefly touched on that one. Some patients, depending on the primary disease and the state of the in which phase of the disease they are, they might need bone marrow harvested cells because there's many of them. There's like the number of cells is actually higher, and that can help the engraftment. The choice is not always based on the patient needs. Also, the donor the transplant center where the donor's harvesting takes place, they might not have the technology or the possibility to harvest cells in from the blood. So it depends on various factors, and of course it depends also on the donor's choice. Not everyone is happy to donate in both ways. Some people are happy to donate with the apheresis, so from the blood, some people from the anesthetics. So we also have to protect the donor's needs, not only the patient's one. Of course, we give the possibility, we explain all the possibility, and then the donor will choose what to do. And ultimately, we do everything to protect the patients, but the donor's choice needs to also be taken in consideration.
Thank you. Question from Zella. How often do donors get recalled to donate again to either the same or a different patient?
It is a very good question. So not very often, but it depends on how common the donor's HLA type is. If you are very, very, very common, bog standard British Irish, which is good, by the way, being very common in this case, it's absolutely great, because of course that means that there's much choice out there. You can choose the best donor for your needs. If the donor has a very common HLA typing, it is possible that they can be recalled for different patients. It's not very common though. For example, it happens if the donor is a CMV positive and young. Why positive? Because usually young people haven't been in contact with the virus yet. So usually the majority of them are negative. Now the patients can be older and the patient are positive mainly. So there is a mismatch there. So a young donor with a common HLA and positive for CMV is really the best. So if you happen to be one of those, you might be called for more than one patient. Of course, it's quite rare. And usually you donate once and you don't donate. But what happens more often instead is that you are asked to donate usually kind of lymphocytes or some cells, like a top up for the same patient you donated the first time. So you donate for the stem cells for the patient, and maybe the patient needs a little bit of a top up, some lymphocytes or some other cells. And because we already know that that donor is compatible to the patient, usually what we do is contact that donor saying, would you mind doing a little blood donation or lymphocyte donation? We call it top up to help the patients engraft and be better.
Great, thank you. Really thorough answers. It's fantastic. So another question from Sadia this is often a bit of a tangent, so you might not know, Dario.
Okay.
I assume you will, to be fair.
I'm not sure.
Is the tissue typing for organ transplants also done in the same way as HLA matching?
I know that. Yes. So at Antonina Laboratory, actually, we do also HLA typing for solid organ transplant because we collaborate with the Royal Free Solid Organ Group. So actually, they live with us. We share the same premises. So, and I've been doing solid organ transplant in my life before. So yes, it's exactly the same. But solid organ transplant, kidney, heart, liver, pancreas, the level of resolution required is less. That means that it is okay to transplant a kidney or a liver or a heart with a lower level of HLA matching. This is because with solid organs, what we need to be more scared about is the rejection. The GVHD is only typical of the stem cells. So yes, the way of typing is the same, but more number of mismatches and also a lower resolution typing is sufficient.
Thank you. That was the end of the questions that we've got in chat. I don't know if anybody's got anything that they'd like to ask Dario, off mute and ask. Peter?
Yes, I just
Hello.
like to say, hi, Dario, just for your information and interest. I'm a kidney donor. I donated a kidney to a friend of mine 15 years ago, and she's still doing wonderfully well, but really amazingly, the matching was all done at the Royal Free because the operations took place at the Royal Free.
There you go. Well, that is great. And I think what you've done is great, Peter, and I'm so glad that your friend is is well. Yes, Royal Free is one of the most important hubs of solid organ and transplant in London. So
and it's because of doing that donation that I wanted to continue to help save lives, that I'm involved with Anthony Nolan and have been for 13, 14 years in different facets of work and voluntary work for the charity. And I continue to do so. It's absolutely amazing.
And we are very grateful for that. Thank you.
Thanks, Peter. And thanks all of you as well. I think I'm just actually looking at all these faces and I'm actually thinking, I wonder how many people's lives have been saved because of the work that Dario does in labs and all the work that all of you have been doing to recruit donors tirelessly over the years. It's quite overwhelming, actually. So thank you. Couple more questions just coming through. Kate has asked about the upper age limit for stem cell transplants. Why do we have an upper age limit?
Anthony Nolan Register is unique because we are science-led, if you want. So we take our decision thanks to our research department that does important research and studies and published literature. And we have found out that if a transplant comes from a donor that is 16 to 30 years old, the outcome is better and the possibility of survival is better. So we decided to invest our money to type the best donors for our patient. That's why you need to be 16 to 30 in order to join our register. Of course, once you join, you can stay there until you are, I want to say 55, 60. Alice, please help me. 60. I can't remember.
You will be off the register. Well, it's changed. I'm looking at one particular person that I know got a letter around their 60th
A letter is 60.
Say thanks, but no thanks.
61, isn't it?
But it's actually 61 now.
Okay.
Yes.
Yes. So to answer your question, basically the upper age limit for a stem cell transplant is that really, because there is a fine line whereby yes, you want to help the register, but again you want to make sure that the donor's health is also looked after. So yes, we wouldn't that. Well, of course, sibling wise, higher ages could be taken in consideration, although it's quite unusual to go above that because, of course, it's still a medical procedure. You don't want to harm the donor, even though it's not really harmful. But but both patient and donors needs to be looked after.
Yeah, sure. I think Jean's thinking of a change in career, maybe. Asking what kind of degree or training should someone do to do your job?
I can do that. I can say that. So in order to do a laboratory scientist kind of entry level in our field, you just need a biomedical sciences degree, so a bachelor in science, the first let's say 3 years. But of course, if you want to progress, then there are other diplomas that allow you to be a clinical scientist. It usually is a 3-4 year course, a clinical scientist in HNI, which is histocompatibility and immunogenetics, which is our field, which I am a training manager for, so helping young scientists to become, through their path to become clinical scientists. And once you're a clinical scientist, a state registered clinical scientist, you are able to authorize and report clinical tests for transplant. So entry level, Bachelor of Sciences and plus, and then of course once you're a clinical scientist, you specialize and you can become supervisor and you can go higher. But yes, that's really the requirement.
So just I love how you say just need.
It is
3 years of study.
It is a very interesting niche of immunology, and it's very, very specialized, and we really, really love it. And so we're trying to give our young colleagues, trying to infuse the love for this for this particular science.
And I'm sure
field.
you do an amazing job, Dario.
Thank you.
So Zella, quick question. Zella has been matched 3 times. Is that unusual?
No, it's not.
Adding to this, that Zella's, Kevin has said that he was matched 3 times, but only went forward on the 4th.
That is
So-
fantastic. If someone is matched to a patient so many times, that means that their HLA type is quite common in the population they live, and that probably means that that's why they are quite appealing for our patients community. So yes, if in this particular field, being common is really good. That means that you can help more people.
Thanks. So not that unusual, Zella.
No.
Any other questions? No, I'll give it a couple of minutes. So just to add, so obviously Dario's been talking about all the science behind transplants, and you know, we're obviously led by science. I do feel that might have some tainted thing when we start saying that now, but we absolutely are. So something that we are looking at as in terms of our recruitment is how we can get more information to make transplants more successful and to make it easier for labs to match. So there will be some communications coming out around how we will start collecting CMV at recruitment, so we can help to make better matches, but we'll provide you with that information as soon as we can. Is there any more questions? If not, I think we can all say thank you so much for, yes, Ella, you absolutely can. Thank you so much for such an informative 45 minutes. I've been here a long time, and I still feel like I've learned things that's going to enable me to put things across in a different way. So thank you so much, Dario.
Thank you very much for inviting me.
Thank you. Thanks, everyone. And it's absolutely lovely to see your